Hello guys, so now we will start with the second episode of the systemic histology. In this we will start gastrointestinal system. So guys, we will first quickly revise so how to identify the slides. If you want to listen the class further, you may. Now we will start. As we can see here, we can see a purple color line at the base and a pink colored layer above. So it is a stratified squamous epithelium. In the same way, we can see here we have a purple color line downward and a pink color above. So it is also a stratified squamous epithelium. And as we can see, we have different layers: first layer, second layer, and a third layer. So it is a GIT. Four layers are there, so we can understand that it is a GIT. And due to the presence of pink and stratified squamous epithelium, the only four parts of the GIT contain stratified squamous epithelium. First is tongue, second esophagus, third oral cavity, and fourth lower part of the anus. It does not have any deep crypts, so it is a esophagus. Next we can see here, as we can appreciate here, we have a dark lower layer and a pink upper layer. So it is a stratified squamous epithelium, and we can appreciate here a spine like structure, which are nothing but a papillae, ear, and a mushroom like. So these are nothing but a papillae, and lower to that we have a skeletal muscles, serous glands, and a lamina propria. So this is a slide of a tongue. By the presence of spines and mushrooms, we can find a papillae, which are nothing but a characteristic feature of the tongue. So next we will identify this one. So as we can see, we have a little finger like projection, which are rugae. Now as we can see here, there are pits which are not extending till the lamina propria. They are just superficially. So by this we can get a clue of a stomach. And another, we can identify that we have a muscularis mucosal layer, and this is a submucosa, and a muscle layer. The muscle layer will be more wider because it contains an extra oblique layer. So it is a slide of the stomach. Next, as we can see here, in the small intestine we have a deep lining, or we can see here we have deep linings, and here we have a deep linings which are reaching till the lamina propria. Here we have a deep linings. So these are all the slides of the small intestine. Now we will differentiate between the duodenum, jejunum, ileum. Duodenum, it has glands which are submucosal glands. These are nothing but a Brunner gland. So by this we can identify it is the duodenum. Next we will go to the slide. It also has more deep projections inside, and it also has a finger like projections. And as we can appreciate, there is no glands and there are no lymphatic follicles. So it is a slide of a jejunum. Next, as we can identify, we here have deep processes, and below that we have lymphatic follicles or a Peyer's patches. So by this we can identify ileum. Main identification point, we have a deep crypts or a villi and a lymphatic follicles. For jejunum, we have a deep crypts and then no Brunner's gland and lymphatic follicles or Peyer's patches. For duodenum, we have deep crypts and Brunner glands present. The deep crypts are present only in the small intestine. Next, in the large intestine, to identify the large intestine, we need to see the goblet cell content, which will be more. So we can see here we have a lot of goblet cells which are appearing a light colored cells. So these are the goblet cells, and the projections are also not that deep, only superficially present, such as stomach, and it has more goblet cells. So this will give us a clue of a large intestine. So it is a slide of a colon. Next, as we can see here also we have a more number of goblet cells. As we can see here, these are the goblet cells. And below to this, as we can identify roughly, this is the circular area, which is nothing but a lymphatic follicle. So by this we can identify it is a appendix. Gastrointestinal system starts from the oral cavity and ends at the anus. We will see the parts of the GIT. First, it starts with the oral cavity that contains tongue, and then it enters the pharynx, then esophagus, then stomach, continues as a duodenum, then jejunum, then ileum. Ileum continues as a larger part of the large intestine called as a cecum, then ascending colon, then transverse colon, descending colon, sigmoid colon, and last we have rectum and anus. And another part which we miss out is the appendix. Now we will see a lining epithelium of the GIT. The only we have to remember is from the stomach till the upper part of the anus is lined by simple columnar epithelium. Rest all is lined by the stratified squamous epithelium, which is present in the mouth, tongue, esophagus, and lower part of the anus. So it is the only trick I can make for you to remember. Simple columnar epithelium is lined from the stomach till the upper part of the anus. Next, we will start with the histology of the tongue. The tongue, it is a conical muscular organ made up of a skeletal muscle, and it is covered by the mucous membrane. As I said, lining epithelium is a stratified squamous epithelium. And now we will see the anatomy of the tongue, which will be asked in the viva. So we can see here a foramen, which is called as a foramen of cecum, and from it anterolateral sulcus is formed, or a groove is formed, which is called as a sulcus terminalis. This sulcus terminalis divides the tongue into anterior and posterior part. The anterior part is larger, which is made up of a 2/3, and a posterior part is smaller, made up of a 1/3. The anterior part of the tongue contains a papillae, and posterior part contains lingual tonsils. Posterior part do not contains any papillae, but can have a taste buds. Now we will see different types of papillae. We have 4 types of papillae: circumvallate papillae, foliate papillae, filiform papillae, fungiform papillae. Circumvallate papillae, these are present just anterior to the sulcus terminalis. These are present only 4 to 8 in number, and just posterior and lateral side of the tongue there is a presence of foliate papillae. These are present only 5 to 6 in number on each side, and rest of the tongue is mostly covered by the filiform papillae, and anterior lateral part and tip of the tongue is covered by the fungiform papillae. And the tongue is divided by a groove into right and left halves. Now we will see a difference between the papillae. All the papillae have the taste bud except filiform papillae. It does not contain any taste bud, and it is only the abundant papillae but does not contain taste bud. Circumvallate papillae are lined anterior to the sulcus terminalis. Foliate papillae are lined posterior lateral side of the tongue. Filiform papillae are present anterior 2/3 of the tongue, and fungiform papillae are present at the apex and lateral margin of the tongue. The point to remember here is papillae are present only in the anterior 2/3 of the tongue. They are absent in the posterior part, and the posterior 1/3 of the part contains lingual tonsils, which forms a component of a Waldeyer rings. Taste bud structure. It is not that important, but may be asked in the viva. So it contains 4 types of cell: sheath cells, supporting cells, receptor cells, and basal cells. Basal cells, wherever we study, it means stem cells. Then receptor cells, these are the main taste sensing cells. They sense the taste and send via afferent nerve fibers to the gustatory center in the cortex. Supporting cells, name suggests it supports the taste bud, and the sheath cells, these are the outermost covering of the taste bud. Now we will see a histological diagram of the tongue. So the tongue, it is a dorsum surface of the tongue, and it is the ventral surface of the tongue. As we can see, dorsum of the tongue has a papillae, whereas ventral part does not has any papillae. And we can differentiate between the papillae. As we can see, it is a spine shaped papillae, which is nothing but a filiform papillae. And we can see it appears as a fungi, which is nothing, or a mushroom. So it is called as a fungiform papillae. And a circumvallate papillae, this also appears as a fungiform papillae, but it contains a taste buds, which I will show you in a subsequent session. And as we can see here, we have a skeletal muscles arranged in a haphazard arrangement. It does not have any regularity. And the tongue contains serous gland and mucous glands. And the lining epithelium, as we can see here, it contains stratified squamous epithelium. Now we will see histological diagram of the tongue. It is lined by the stratified squamous epithelium and it contains taste buds, as we can see here, and a light zone within the epithelium, as we can appreciate here. It is a taste buds, and below this we have this lamina propria, and as we can see the skeletal muscles are arranged in the bundles, and these are the serous and the mucous glands. Now we will see a proper image of the taste bud. This is a circumvallate papillae which has taste buds at the lateral sides of it. So as we can see, this is the epithelium, and within the epithelium we have a taste bud which are appearing as a light zone. So these are the taste bud. Identification points we can identify easily by looking at the spines, which are nothing but a papillae, and we can see skeletal muscles and taste buds if we appreciate properly. Now we will see the layers of the GIT. GIT, it is has a 4 layers: mucosa, submucosa, muscularis externa, and serosa. It is also called as adventitia. Submucosa and serosa, these are mainly made up of a connective tissue. Mucosa, it has a 3 layers: epithelium, lamina propria, and muscularis mucosae. Epithelium, as I have stated already, the 4 parts are lined by stratified squamous epithelium, which are oral cavity, tongue, esophagus, and lower part of the anus. Rest all is lined by the simple columnar epithelium. And lamina propria, which is also made up of a connective tissue, contains blood capillaries, lymph vessels, and nerves. And muscularis mucosae, it contains a smooth muscle layer which separates a lamina propria from the submucosa. Now we will see submucosa. As I said, it contains a connective tissue which contains blood vessels, lymphatics, and nerve fibers. We need to remember 2 peculiar points. The submucosa has a part of an enteric nervous system. What is enteric nervous system? It is a part of an autonomic nervous system. It runs involuntarily and controls the GI motility, secretions, etc. So it has 2 parts: submucosal or a Meissner's plexus, and another is myenteric or a Aubach plexus. The submucosa contain a part of enteric nervous system, which is submucosal or a Meissner's plexus. So we can remember this by submucosa has a yes. So Meissner's also has a yes. So submucosal plexus or Meissner's plexus. And another peculiar point to identify the duodenum. The duodenum has a Brunner's gland in the submucosa, which are nothing but a mucous glands. Muscularis externa. It is made up of 2 layers of the smooth muscles, a circular and a longitudinal. The circular layer, it is the inner layer, and the longitudinal is outer layer. And muscularis externa has a second part of the enteric nervous system, which is myenteric or a Aubach plexus. And here we have 2 peculiar points to remember. There is an extra oblique layer in the stomach. As we have seen, there are only 2 muscle layers, circular and longitudinal, but the stomach has a third layer which is called oblique layer. And next we have in the colon, the longitudinal layer, circular and longitudinal. In the colon, the longitudinal layer forms 3 bundles of muscles which are called as a tinea coli. It is a gross identification point of the colon. We will see it later. And serosa is made up of a connective tissue. Now we will see a schematic diagram. As we can see, it is the lumen of the GI part, and surrounding it we have epithelium, which is a simple columnar epithelium. Below this we have a lamina propria and a single layer of a smooth muscle which is called as a muscularis mucosae. And around it we have a submucosa, which contains blood vessels, nerves, and connective tissue. And as we can see here, in the submucosa we have a plexus, which is a submucosal or a Meissner's plexus, which has only parasympathetic part. And next we can see this is the circular layer of a smooth muscle, and around it we have a longitudinal muscle, which both constitute a muscularis externa. In between the circular and the longitudinal muscle layer we have another part of the enteric nervous system, which is myenteric or a Aubach plexus, which has both parasympathetic and sympathetic part. And outer to that we have a adventitia or a serosa, which contains connective tissue. Now we will start with the histology of the esophagus. It is a long, narrow muscular tube that extends from the pharynx till the stomach. So it helps in the swallowing of the food. The mucosa is lined by the epithelium called stratified squamous epithelium. Submucosa contains esophageal glands. Muscularis externa has an inner circular outer longitudinal layer, and as we move down through the esophagus, the smooth muscle content increases. We have seen same thing in the trachea. There also the smooth muscle content increases as we go down. Serosa is made up of a fibroconnective tissue. Now as we can see, we have a stratified squamous epithelium. Below this we have a lamina propria, then we have a muscularis mucosae, then we have submucosa. These are the esophageal glands, and we have muscularis externa, and at last we have a adventitia or a serosa. And as we can see in the stratified squamous epithelium, we does not have any grooves. It is a continued line, so it will make a differentiation from rest of the GIT. The rest of the GIT contains many grooves which is absent in the esophagus. So now we will identify in the microscopic view. As we can see, there is a epithelium which has a multiple layered of cells. So it is a stratified squamous epithelium and it does not has any grooves. So easily we can identify it is a esophagus. And other than that, we can identify by looking at the 4 different layers of the GIT. So first we are seeing a mucosa which has a lamina propria, and we have a thin muscle layer which is a muscularis mucosae, and below this we have a connective tissue which is a submucosa, and we have more muscle bundles called muscularis externa. It has a circular inner layer and a longitudinal outer layer. Now how we will identify the esophagus is, we should see the stratified squamous epithelium. It will appear as a pink colored upper part and a purple colored lower part, so we can identify it easily. And has 4 different layers of the GIT, and it has no finger like projections. It is the main peculiar point to identify esophagus from the rest of the GIT. Now we will start with the histology of the stomach. The stomach, it is a hollow muscular organ. It is also called as a food bag, as it temporarily stores the food within it. It receives food from the esophagus and converts it into the chyme inside the stomach and sends it to the duodenum. It has 4 parts. The first cardia, then fundus, then body, and a pylorus, or antrum. Now we should remember the cells of the stomach, where they are present. Fundus and body has a parietal and the chief cells. Pylorus has a G cells and a mucous cells, whereas a cardia has a mucous cells. Most important to remember is fundus and body has a parietal and the chief cells. Pylorus has a G cells. This is the most important to be remember. On cut section of the stomach, we will see a folds which are called as a rugae. We can see with our naked eyes. Now we have a different types of cells in the stomach, and we will see their functions, which will be asked as in a viva. Parietal or oxyntic cells, they secrete HCl and intrinsic factor. What is its function? HCl helps in the digestion of the food particles and activation of the pepsinogen enzyme, and intrinsic factor, which is required for the vitamin B12 absorption. Deficiency of intrinsic factor can cause pernicious anemia. Chief cells or peptic cells, they secrete a pepsinogen. It is a proteolytic enzyme, means it digests the proteins. On HCl, it converts into pepsin. And we have a mucous cells which secrete a mucin or a mucus for protection, and G cells which secrete a gastrin. Gastrin is needed for the secretion of the HCl from the parietal cells. Now we will see the 4 different layers of the stomach. Mucosa has a epithelium, as I said, from the stomach, lined by simple columnar epithelium. So it is lined by simple columnar epithelium, and I said esophagus is the only part which does not have any infoldings, and rest all of the GI has a infoldings. So we can see it has a dips into the lamina propria that forms gastric pits, into which gastric secretions are secreted. Lamina propria, which is made up of a connective tissue. Muscularis mucosa made up of a smooth muscle layer. Submucosa, it has a irregular connective tissue, blood vessels, lymphatics, and plexus of nerves. Muscularis externa. Here we have a 3 layers of the muscle. Rest of the GIT has a 2 layers of the muscle, circular and a longitudinal. But it has an additional layer which is a oblique layer. So it is oblique layer is lined completely inside. Serosa is made up of the connective tissue. Now as we can see here, we have many folds, so it is a other than esophagus, and we can see we have a columnar epithelium, simple columnar epithelium, and we have here parietal cells which will appear a dark pink colored, whereas chief cells will appear a light colored, and we have here muscularis layer. Now we will see the histology diagram of the stomach. As we can see, we have a pits. So this is nothing but a gastric pits in which gastric secretions are secreted. And we have a columnar epithelium, and we have glands below the epithelium in the lamina propria. And we have a pyloric glands, and this is the submucosa. It contains connective tissue, and we have a muscularis layer. We will identify the stomach by looking at the gastric pits, which will not go too deep inside. But whereas when we see small and large intestine, they will have a deep invasion into the lamina propria. So as we can see here, it is a lamina propria layer. So in the stomach we have only a little depth, but whereas when we will see in the small intestine, it will contain a large deeps. So we can identify this one as a stomach. Below the gastric pits, it has a gastric glands which will appear as a pink and a white colored areas, and we will can see appreciate 4 layers of the GIT. How we will appreciate 4 layers of the GIT? It will appear in a different colors, and pyloric glands will be seen if present. Histology of the small intestine. It is a tube that extends from the stomach to the ileocecal junction. It is the principal site of the digestion and absorption, because most of the enzymes are released here and absorption takes place here, except water which is absorbed in the large intestine. It has 3 parts: duodenum, jejunum and ileum. When we see with our naked eye, we can see certain folds which are finger-like projections called as a villi. In stomach we saw a rugae which are seen with a naked eye. In the intestine we can see villi. The downward projections into the lamina propria are called as a crypts. So these are the long than the stomach and large intestine. Now we will see here the projections. These are the finger-like projections which are coming above the level of the epithelium. These are called as villi, and within it the part which goes downward, this is called as a crypts. The epitheliums, the epithelial cells, this is a simple columnar epithelium. It has a many cilia-like projections. These are not cilia, these are microvilli. 4 different layers of the intestine. The epithelium, which is lined by simple columnar epithelium all over and has a goblet cells, but they are not much. These are only very less in number. Lamina propria. It shows Pierce patches. It is an important identification point of the ileum. Pierce patches are nothing but lymphatic follicles. And we have muscularis mucosae. Submucosa contains loose connective tissue, mesenteric plexus, etc. as we have seen. And the most peculiar point is Brunner's gland, which are nothing but mucous gland, which are present in the duodenum in the submucosa. Muscularis externa. It has 2 layers of smooth muscles, and mesenteric plexus serosa has a connective tissue. Rest all is same. What we have to remember here is it has a low goblet content, and it has a Pierce patches in the ileum and a Brunner's gland in the duodenum. These are the peculiar points to remember. So we will differentiate by this. As now we can see by the diagrammatic representation, we have finger-like projections which are called as villi, and we have downward projections are called as a crypts, and which is lined by simple columnar epithelium. We have a lamina propria, we have a muscularis mucosae, and as we can see here, these are the mucous glands which are nothing but a Brunner's gland. And below this we have muscularis externa, and below this we have serosa or adventitia. That was a diagram of the duodenum because it contains Brunner's gland. And now we will see the diagram of the jejunum. Jejunum, we have finger-like projections and we have a crypts, and it does not contains any submucosal glands as we can see here. which are absent, and it also does not contain any pair patches, so it is also absent, so it is a jejunum. And we can appreciate a single layer of muscularis mucosae, we can appreciate muscularis externa and adventitia, so it is a jejunum. Ileum, it also has a finger-like projections and a crypts, and it has a lymphatic follicles in the lamina propria. I am repeating it, lymphatic follicles in the lamina propria. So it is the most peculiar point to identify ileum. The lymphatic follicles are also present in the appendix. We will see that later and compare that. And then we have a single layer of a muscularis layer, then we have a submucosa, then we have a muscularis externa layer, and we have adventitia. Now we will see under the microscope and identify which slide easily by looking. So as we can see, it has a two large depths. So it is a small intestine, it has a large depth, it also has a large depth, and it also has a large depth. So we can identify it is the intestine, because stomach does not have this depth, it has a very short depth. And if we identify properly, we have here glands which are present in the submucosa, which are appearing as a white colored area. So these are nothing but a Brunner's gland, which are seen in the submucosa. It is the most peculiar identification point of the duodenum. Next we will see ileum. Ileum, it has a finger-like projections, so it is a small intestine. Then it has lymphatic follicles or Peyer's patches in the lamina propria, so we can identify it as a ileum. This both Brunner's gland and lymphatic nodules or Peyer's patches are absent here, so we have a large finger-like projections. And in the submucosa or the lamina propria, we don't have any lymphatic follicles or Brunner's gland, so it will become jejunum. It is the simplest way to identify. And as now I can see, it is a diagram of the large intestine, mainly the colon. So we can identify it also has a large finger-like projections, but the depth is not that deep. And if we see properly, it has a many white colored dots, which are nothing but a goblet cells, and goblet cells are abundant in the large intestine, because the large intestine needs to pass the stools easily, so needs more mucous secretions. The We cannot appreciate that much properly a mucus glands in the small intestine. It all, and we can see this is the tinea coli. As I said, the longitudinal muscle layer modifies into bundles. This is the tinea coli. We have three. We will see it later. Now we will start with the histology of the intestine. It extends from the ileocecal junction and end at the anal canal. It includes cecum, appendix, colon, rectum, and anal canal. The main function is absorption of the water and formation of the solid stools, because digestion and absorption of the food takes place in the small intestine, and absorption of water and formation of the solid stools takes place in the large intestine. It has some non-harmful bacteria which are good for our body, which helps in the production of vitamin K, vitamin B12, and they also prevent us from the pathogenic bacteria. So colon has a three characteristic properties. This will be asked in your gross viva, practical, and histology viva. So these are tinea coli, circulation or ostration, and appendices epiploicae. Tinea coli are nothing but longitudinal muscle which is present in the muscularis externa layer. It forms three bundles which are called tinea coli. As we can see here, a longitudinal muscle layer. This is nothing but a tinea coli. We will have three, but we are appreciating only one. Another may be present in this side on the posterior aspect. Then circulation, as the name suggests, circulation means sac-like. It is also called as ostration. Sac-like structures are formed because the longitudinal muscle is shorter compared to the length of the large intestine. So that causes the formation of the sac. As we can appreciate these are the sacs. These are called as circulation. These are only present in the colon. Appendices epiploicae. These are nothing but a peritoneum which forms a pouch inside which a fat is filled. So they appear as a yellow colored structure. If you have seen on the dissection, the yellow colored substance over the large intestine or the colon. These are the most important peculiar points: tinea coli, circulation or oscillations, and appendices epiploic. Now we will start with the colon. The colon, as I said, it is lined by the simple columnar epithelium. It has a simple columnar epithelium and has many goblet cells. It is the main point to differentiate it from the small intestine, and it contains lamina propria, muscularis mucosae, submucosa, and maybe few lymphatic follicles may be present, but mostly they are not present. And we have inner circular layer, and longitudinal layer is modified into tinea coli, as we can see. Now, as we can see here, we have a finger like projections, and we don't have much deep invasions, and we have a many goblet cells. If you see properly here, we have a many goblet cells, and we have a muscle layer, submucosal layers, and a longitudinal layer. So we can identify the large intestine by seeing crypts are not going deep, and epithelium has many goblet cells. Many goblet cells, it is most important to identify, and tinea coli can be seen if the presenter has kept the tinea coli, and we can appreciate 4 different layers of the GIT. And now we will see the last part of the GIT, it is appendix. Appendix, it is smallest part of the GIT. So it has a lining epithelium of simple columnar epithelium. It contains lymphatic follicles. As I've seen in the ileum, it also contains lymphatic follicles. How to differentiate between ileum and appendix? We can identify by the presence of goblet cells, because large intestine contains many goblet cells. It is only point to differentiate. As we can see here, it is the gland which has many white color dots, which are nothing but a goblet cells. We will see later. And we have a submucosa and a muscularis mucosa, muscularis externa, and surrounding it we have a serosa. We can even appreciate the lumen if we see properly. For identification, we have lymphatic follicles, so we should not get confused with the ileum, because ileum also has lymphatic follicles. The lymphatic follicles are present only in the two parts of the GIT. These are appendix and ileum, and few can be present in the colon, but mostly they are not present. So main identification point is goblet cells, which will appear as a white colored dots, and rest all will be similar. Identification points, here villi will be absent. As we can see, there are not many projections, so villi will be absent and has numerous goblet cells, and lymphatic nodules are seen in the submucosa, and we have 4 layers of the GIT. So thank you guys for watching a complete video, and the video has become a little longer, but I think it was useful for identification points. So for the notes, you can visit my Telegram channel, which I have provided in the channel's bio, or I will write a channel name in the description box. And if you love the content, please like, share, and subscribe with your friends, so they may get benefited, and it will also motivate me. Thank you guys.